Yeah, so welcome everyone to the Breakout Session number 3, where we have the topic of spatial methods, especially computational methods for spatial omics data. It's going to be me and David who will be leading this discussion. And we thought it would be like a nice idea to just like introduce ourselves because we might not be as famous as Fabian and Bo, for example. So maybe you want to start with just like introducing me.
Yeah, I'm David. I work as a postdoc at the Broad Institute and the Eric Management Center with HCA. And in the context of this session, I have research interest in system communication methods, especially informed by spatial data. That's what my best interest is.
Yeah, and I'm Alma Anderson, and I work at Genentech at the AI/ML department based in San Francisco. So I'm very happy to be here after an 11-hour travel trip. Apparently, you need a visa to enter Canada, which I learned. So I had to stay overnight at the airport and take a later flight, but I made it in time. So that's great. My interest in the context of this kind of meeting or like this session, I would say, is like context-dependent models, looking at sort of like how the extrinsic state of cells maybe influences their intrinsic state. And of course, was like common coordinate frameworks for spatial omics data is something I'm pretty passionate about. And so we'll start it off by just going through like our ambition or like our agenda for this session. And so what we really want to do here is not to like find solutions to all of our problems, because I think that's quite ambitious, but we would like to try to chart the computational problem space, trying to understand what challenges that you guys are facing when working with spatial omics data, especially with respect to the computational methods. And then once we've sort of like charted this space, hopefully we can identify and prioritize the current challenges and like which ones that are most urgent to address. And we'll try to focus our discussion on the HCA community, since that's kind of like what we are all here for. And so we sort of like decided to stratify the discussion based on sort of like the areas along this study life cycle, focusing on computational methods for experimental design and also the analysis of our data. And we kind of like made this flowchart when trying to structure this session. And I thought I'd just like briefly go through that, like how we reason when setting up this discussion. So first of all, we sort of try to say that, well, we should identify what like the most important and general questions that we are interested in are, both now and in the future. And of course, then like whether spatial omics data is necessary to answer this question. And if it's not, maybe it's a really good question, but it's not really for this session. And if it is like necessary to use spatial omics data to answer these questions, we can sort of like ask whether the experimental design of these sort of like methods or experiments are straightforward. If it's not, then we sort of like will save this for discussion A, which they will both be more into soon. And if the experimental design is straightforward, we can sort of then look at whether we need additional or more complex computational analysis to answer these different questions. If that's the case, we can see sort of like whether, if it's not, I guess we're not really going to discuss this during this session either. If it is, then we sort of like see whether computational methods already exist or whether we need to like invent or come up with new ones. That's going to be discussion B. If there are methods that already exist, we're going to sort of like see and assess whether we think those are satisfactory or perform up to our standards. And if it's not, again, we can put that into discussion B. And if there are already existing methods, we can like add this to this list or repertoire of methods that we can all use, because we don't really have to reinvent the wheel essentially. Okay, sure. So hand it over to David to do the discussion.
Thanks. Yeah, so for the first part, and again, so we'll moderate the discussion, so hopefully we hear a lot of ideas, and this is just like to structure this a bit and help us write up the output of this. So for experimental design, overarching themes probably are do we foresee challenges that persist despite the advancement of technologies? So how far can we push this? How do these challenges translate over different technologies, right? Think about targeted, untargeted resolutions. What do solutions and implementations look like? Now, some ideas for you to comment on after we finish this introduction. So we have two example problems here in this space of experimental design. The first one is panel design. So for targeted spatial techniques where we need to design gene panels, for example, let's Murfish, for example, how do we assemble these panels? And then questions that relate to this that we could discuss are what are we optimizing for, right? So number of cell types, including rare cell types, particular targets that we could maybe impute from the panels that we assemble. Do we optimize between cell type and cell state variation for one or the other, depending on a particular scenario? How do we come up with these panels, thinking about testing RNA-seq references? And then out of the methods that exist here, which ones are we happy with? Do people have like practical experience? Yeah. Second example question, power analysis. So not just for targeted assays, but in general, if you think about tissue motifs, how do we think about defining cohort sizes to assemble actionable statistics? For example, cell type abundance, microstructure prevalence, think about cell neighborhoods. And I think in the space of spatial data and power analysis on spatial data, it is particularly difficult to define what you're looking for. So are you going to do a power analysis on a particular neighborhood constellation that occurs with a particular frequency? So do you need a pilot to define that constellation?
It was the thing that volume.
Yeah. I thought that's what you got.
Yeah. So yeah, do you need like pilot experiments to define the setup of the power analysis? So yeah, think a bit about this, and then we'll come back to this at the end.
Right. So let's see. I think you guys should be able to hear me. And so for discussion B, we're going to focus a bit on the computational methods for analysis. And so just sort of like within the realm of this discussion, we really think it's important to try to identify the main bottlenecks for analysis and interpretation of spatial omics data. And a good way to sort of like think about this framework, or like during the discussion, is, for example, in the context or like in the way of like expected input or output. And just to exemplify that for these sort of like spatial deconvolution methods, which I would sort of like explain a bit more what I mean with very soon, we can sort of like say that like the expected input would be like the annotated single cell and spatial omics data sets, and the desired output would be the proportion of each cell type at every spatial location. And if applicable, it's also nice to just like try to keep in mind how existing methods could be improved if there are already existing methods for this. For example, is the current limiting factor for these methods to be satisfying the performance? Maybe we don't get like the results that we expect. The user interface, maybe it's hard to use. The code base might be deprecated. Scaling issues, maybe it works super well for a small data set, but then for the larger data sets, it's kind of like a big issue, or something else. So these are just like things that are good to keep in mind when we sort of like move on towards this discussion. And sort of like in line with the previous part, we thought it could be nice to give some examples of problems that we think fit into this discussion, but you're obviously welcome to like add other problems, or like if there's something that we haven't considered here. And so the first one would be this sort of like idea of decomposition or deconvolution of mixed spatial omics data. And I think two good examples here would be like the Visium or slide-seq data. And so maybe a lot of you are familiar with like the idea of spatial deconvolution, but just like reiterate that and make sure like we all understand the concept. So for several of these sort of sequencing-based spatial methods, they don't really provide single cell resolution, but instead we can like retrieve this mix of signals from multiple different cells that contribute to our observation. And we somehow want to like demix this, and actually we want to sort of like force this into sort of like being more informative about cell types rather than use like gene expression programs or factors. And just to illustrate that concept, I can like add this image here. We can look at this mouse olfactory bulb. It's a schematic, but essentially we don't really know the cell type proportions. All we have at our disposal is the gene expression. We somehow, maybe using single cell data, want to decompose this into actual proportions, telling us where the different cell types are located within our tissue. Another sort of like, and I guess some questions that relate to this will be listed here. And it might be a bit tedious to go through every single one of them, but as some examples, like, is this a solved problem? I think every week, at least at Penentech, we have this kind of like a journal club where we focus on spatial omics methods. And I think I add 1 or 2 methods for this every single week. So a lot of people are working on it. Like, are we considering it solved, or it's something we should still continue to pursue? And also, will this be an issue in the future? Maybe like we will have more of these single cell resolution-based methods. So we might not even use this in a slide-seq. So is this something we should invest time in? Another example is this idea of common component framework, something I did a bit of work on during my PhD. And it's kind of like related to when we speak about common component frameworks for single cell RNA-seq data or single cell nucleide data. But also here we have this additional factor that we need to take into consideration, being the spatial coordinates. And so we kind of like want to map things both in the gene expression space and in the spatial space together, allowing us to compare different signals across both time and space, for example. And I think Nadav gave a really sort of like good example of this in his famous Atlas presentation previous today. And just again to illustrate this concept, we have observed data here, which we might collect from multiple different individuals. We want to align that to some reference, and then we can actually compare the gene expression or protein expression across these different individuals, allowing us to make this like differential spatial analysis as well. And then, of course, we have this issue of integration between multiple platforms and modalities. And so I think there's like an even more diverse landscape of methods when it comes to collection of spatial omics data. And this is something we really want to correct for, or like take into consideration when we're working with our spatial omics data. And so one really important question there is, like, are the single cell methods sufficient, or should we try to like develop spatially aware methods? And finally, as for like the example number 4 here, we can focus on cell-cell interactions, which is like this very unique feature that we can almost like only obtain from spatial omics data. And so, of course, like we know that if two cells are close to each other, they might interact, but it's not necessarily such that they interact. And so how do we actually model the true interactions and not just base our assumption on proximity? And also, do we want to rely on databases based on like receptor and ligands, or do we want to sort of like do some de novo identification of interactions? And so those are also like discussions and examples that we would like to like engage in when we hit this like discussion point. And with that, we just want to mention briefly what we sort of like had in mind to deliver after this discussion is done. So I hand over to you.
Yeah, so to force us to make like facilitate a discussion that doesn't just meander a bit, we thought we would think a bit about problems that will be raised now. Yeah, we would try to categorize them into this A or B discussion, so more like experimental planning or more like analysis, whether this is something, or that is a question that requires a new method, or that is something that is partially already addressed. If it is partially already addressed, like to what degree is it satisfactory, right? Like, is there like tweaks to the way that question is framed in maths, or is there like tweaks to the implementation that are necessary? Who's interested in this, right? Like different spatial questions are relevant for different bionetworks. So here, yeah, ideas of like stakeholder sets, so like people in bionetworks, or people in model development communities, or yeah, people in other communities that want to like potentially dive into spatial data but don't quite know how yet. Yeah, and maybe like how urgent these problems are for the next steps of human cell atlas. Yeah. So an output is probably also a good way of thinking about a bunch of these problems. So like if you have things in mind, like think about like how that in an output like differentiates your thought from something that exists. So like how that coincides with something else. How methods provide added benefit to what exists already, right? Like especially with spatial, right? Like what is added on data from dissociation methods, yeah, and then stakeholders. Yeah, so this is the summary slide. We will leave this only up. Again, just for ideas. So we have these 2 points for discussion A and 4 points for discussion B. If you feel to add more thoughts that you have, yeah, and then yeah, we essentially just invite any comments and we try to moderate it a bit. Yeah, so maybe
maybe we can also say like you want access to the slides, you can just like scan the QR code. It might be helpful to just like outline the different discussions and topics a bit more. But we'll start with discussion A, I think, and we'll like assess whether that's something you're interested in discussing more, and then move on towards discussion B. So maybe one sort of like this general question for the audience. So like if you're facing any challenges or issues with like the experimental design, and so like how you're approaching that currently, and where you think it could be like areas of improvement essentially. So I think that's something we could throw out there and start with maybe. And comments on this like super welcome. Yes.
Hi, Sean at University of Michigan. So, I mean, from the experimental design standpoint, for me, the biggest challenge is how do you take spatial data across a panel of samples, or in my case, patients? How do you take spatial data across a set of patients and generate different outputs from each patient to actual phenotypes? So that's a huge challenge. Probably way beyond but it's a big challenge for the field. going forward how to take what we give in the spatial context.
Could you elaborate briefly? Maybe with an example.
Yeah, so an example, I'll have let's say I have a Visium profile from one patient and a Visium profile from another patient. Let's say patient A responds to some therapy, patient B doesn't, right? And let's say you have enough of that you have power where you could actually detect the difference in patient response to a drug. How do you take the individual profiles and create them together? You can imagine going through like a single cell like pipeline and seeing some of the differential expressions, but it would still need to go back to the spatial information. See, is there anything in the spatial data that would say response to the drug? Or something like that.
So what I hear is, for example, yeah, so there's this issue of like defining like the motives in spatial data, right? That you would start correlating. Yeah, I think there's definitely a couple problems there.
But it's probably bigger than just.
I think it's sort of thinking about like the future of where spatial will be in terms of this. So it's just something to throw out there for people to keep in the back of your mind. So how are you going to get to this do something.
Yeah.
Yeah, super cool. Yeah, so then you have one online question.
I think that goes to your power analysis point. So I'm just curious, what have you guys or anybody thought about how you calculate those? How you calculate power? What numbers of cells? What numbers of individuals? Like what is that in this space right now? It's more clear for the single cell data, but I'm curious if there's any clarity in this space.
I think there's some thoughts on how much data you would need to see to observe differences in pairs of neighboring sets, for example. Like there's also some relatively direct statistics you could do there, right? Like if a particular neighborhood as defined by a pair of neighboring sets is perturbed as those not being neighboring anymore because one is lacking or like there's somehow less diffuse in the tissue or more diffuse in the tissue. I think that's the key idea that I'm aware of.
I can also add to that, and with some bias here, maybe like I can think of two papers, like one by Baker and one by the Bode-Millen group, where they look at, for example, how many fields of view you need to collect in order to identify these or like a set number of cell types. I guess, or like the more data you collect, like the more cell types you're gonna see. But when you start to see like a saturation, like you seem to have captured the diversity of the cell type landscape. And they can sort of like see whether like you need to collect a lot of large FOVs, for example, or like multiple smaller ones covering different regions, right? The other one would be sort of like, as you mentioned, with the interaction analysis. And I guess the third way of thinking of this is kind of like the macro structures, where it's not just like cell types, but you have like maybe spatial motifs that you try to identify within the tissue that kind of like relates to like this idea of capturing, so like different motifs using a set number of FOVs, for example. Does that make sense? Yeah, so I think those are like the main aspects that people have explored so far. Yeah. So yeah, let's do the online question. So this is from Vitali. Hi, Vitali. So he says here,
Hi, Hi,
what do you
hello.
mean by sold? Okay. Wait, maybe you want to
wait.
speak for yourself. Yeah.
Yeah, I think that thing doesn't really show what I want to say. I mean, I wanted to add on what you were discussing just now, power analysis problem. I think that I agree with the take that it's useful to find certain tissue neighborhoods, but maybe taking a step back, a simpler way would be to look just at H&E histology sections of the tissue and see how well represented the areas of interest are. For example, in our paper on cell location methods, we used special data from the human gut that contained lymphoid, tertiary lymphoid structure, and it contained just one. While overall there might be, let's say, thousands of of special observations. The observations that represent your area of interest can be in the count of a few tens. And I think that's maybe one way, relatively easy way to assess this is to look at histology sections before doing special experiments, and just select which sections are likely to represent all of the areas of interest. We are also doing similar work with Mus Hanifa, as in Baraktar group is doing similar work with Mus Hanifa on the human embryos, where a kind of histological analysis prior to spatial profiling is a big part.
Nice. Thank you for that comment. That's super helpful. Did you also want to read your question in the chat, or do you feel as that's been answered?
I mean, that question is more general. I like how you structured the review of methods, and of does the inputs match and so on. But what is a bit missing is how do you define whether a problem has been solved? And it seems to me that some problems were indeed solved, but there are many nuances of where, let's say, analysis of Visium doesn't really show us what we want, or where the methods don't scale to some other technologies. And it's useful to discuss what exactly, to make a public list of what was done and what not solved completely.
Yeah, I think you raised a super important point there. So we kind of need frameworks to evaluate and so assess, and also just agree on what is sufficiently good, because you can always make incremental improvements, right? But in the end, so you kind of want to focus your attention more urgent needs maybe. So yeah, that's
what I also mean is that there are kind of conceptually distinct sub problems that need to be solved and weren't addressed. It's hard to come up with an example now. I mean, maybe one example would be analyzing high resolution spatial transcriptomics data like stereoseq without actually aggregating it into bins to make it more like Visium. We don't have an intuitive way to segment the data, identify cells.
Yeah. There first? Okay, yeah.
So my question was also related to power analysis. So when you define, in the Human Cell Atlas, when you define cell types, how do you take into account, cells have activation states, right?
Pilot study, right? And also maybe to use comment a bit more on this whole idea of the power analysis step. It could also be so that maybe our main objective should be to establish a framework for executing the power analysis, and then the people that have the biological expertise, which is definitely not me, can actually then look at the different cell states and types, and plug that into that framework, right? So you can separate those two different processes. Does that make sense?
Yes, yeah.
Okay, cool. That's awesome. So we're gonna move on to Giovanni. Has a question?
Yeah, I have a question related to this. Most of the work that I'm familiar with, where spatial omics data has been used effectively to predict a phenotype that would not have been able to discriminate based only on singles, on the non-spatial structure. Is work from Odemiller and Pelkmans in Zurich. There's been work also from some labs here in Canada. I think there were 2 natures back-to-back on lung and brain cancer early this year. And so all this work has been looking mostly at cell type neighbors. So basically, the input of a classifier is the cell type label and the cell type labels of the neighbors, right? And then you want to discriminate health or disease, or 400 slides of codex or so. Do you think that there are other type of, let's say, biological entities or things of ways or other ways to think of the biology underlying that's not cell type specific that helps in discriminating the phenotype? So do you think that just looking at the genes in spatial structure would be as powerful as for predicting specific phenotypes that is not cell type dependent? So in other words, do we need cell types in the spatial structure for these discriminant phenotypes, or maybe just the measurements, the molecular measurements, or the images, and so on and so forth are enough?
Yeah, that's a super good question that like I'm struggling with every day. I think like you really need to label all our data, right? And it's not like a straightforward answer, but I think one really good thing to keep in mind is that you kind of like need to interpret your results in the end, right? And currently we kind of like tend to develop spatial methods or computational methods for the spatial data, and you present those results to someone who's like a domain expert, for example. And then I think it might be harder to just like present a like gene expression set or just like a factor. Like speaking in terms of cell types makes it way easier for like a lot of people to interpret and sort of like understand the data and the underlying composition of those macrostructures. And so I don't think like necessarily that we have to label each and every single cell in our spatial omics data, but it might guide us and aid us in the process of understanding our results. That would be my comment on that, but maybe David, you have something to.
Yeah, maybe just like throwing a thought back. I guess one of the expected advances now coming in with like the very high resolution phenotyping of cells from SIM7 and ASIC is that you can like map out these like much more fine-grained changes than you'd expect in cancer, which all of these are cancer papers, right? Like better cell dysfunction. Yeah, I guess a lot of like early fibrotic changes. So I guess that using these spatial methods, you could then also map out these gradual and fine-grained changes onto spatial structures and then characterize stuff there better. But that then relies on the stuff that's going on the dissociated data right now. So I guess it would be natural for this to come about later. But yeah, I do think this then comes up in these other disease areas. Yeah, but yeah, maybe like, yeah, maybe it's a good thing to discuss like how that differs between yeah, work on cancer stages and sort of like gradual disease aberrations that often like involve like relatively fine-grained states. I don't know if you have thoughts on that.
Yeah, I think it's a really important point that our analysis of statistics. In a way, like I always feel that we don't really have a lot of statistical tools to handle. We don't know how to handle like multiple donors. We have a problem with kind of inflation of cells and single cell data in spatial data, where we can from one tissue extract 1 million cells, and then automatically your artificial statistical power is inflated, like to an extent that any perturbation test that you would do would kind of like show something that doesn't necessarily exist, and also kind of like our spatial statistics, kind of like Ripley statistics and these like models that people are generating in the past are not good enough because their assumption is kind of like inferring a kind of like a random structure, which is not true in all tissues. Actually. Like all tissues have like a specific organization. So I kind of like really need to kind of like have like a statistician superhero, like love or something like that. That's can kind of like answer these very fundamental questions. That because in the end, I think that we often kind of tend to the fact that like the spatial method, that spatial. There are a lot of good spatial papers, but a lot of papers in the end, like you show here, this is what we see. This is this cluster of cells that are coexisting together. What does it mean? Is it statistically significant that these two cells are actually together in space? What does it mean? Like there's no statistics, there's no kind of like counting for multiple samples, for multiple donors, for the effect size, for the my English intelligent, linear like mixed effects model, like kind of like approach, which I think is really desperately needed. Like the neighborhood size, all these things need to be kind of like inputted, kind of like a model that will be able to tell us like to gain like true statistics. I always feel like in spatial, and also the fact that some of the spatial technologies are grid, other like that completely changes your statistics. Like in your kind of like typical neighborhood size, it's like then into specific structure, then it's discrete data, it's continuous data. I just feel that all of these questions that are maybe the most important ones as researchers in the end, like after we kind of like, yeah, spatial technologies are cool, we've all seen it, it's really nice, we can do cool things, but now we want to kind of do science. Wait.
Comment over there. Yeah.
So this question is about the panel design. So I looked at the 3 technologies, commercial technologies. Panels are totally unsatisfactory. I don't know how they designed it. They must have had some logic. But when you try to study specific problems, panels are. So I think there are 2 solutions. One is to go big, you know, scales to say 6,000, 10,000 genes. Pretty much a lot of biological and relevant things. Or the other thing is to go specific, which would mean custom panels for each organ type, which, you know, human cell atlas can help come up with. Because, you know, then you may be able to study 1,000 genes. When you're studying only liver disease, then you need your own panel. And at the moment, to get custom panels working commercially is
Just to follow up on this, it's really nice to have this discussion here. For atlas design, we really need to 3D spatial. So as I mentioned, we've been looking on what we call organ-based antibody. And those are codecs, and SIMS, and I don't know how many of you are actually using those techniques.
Kind of good.
Okay. So in hardware became only about. So that's a different. But for those healthy organs, we have 13, 20 organ-specific and isolation methods. So we are trying to make sure that all of these omics also have vasculature markers and hepatic markers. So if you are interested in studying healthy organs, look at those charts. It takes a lot of time to generate them and consensus. But then if you use them, it's automatically aligned also with the other. Just like if you run Asimus, it's also aligned with the Asimus. There are these 2 ways you're actually getting data that's already in the state. So happy to talk more about this, but this community also benefits from what you have done. Not by me, but by a lot of other people.
That's super interesting. Yeah, we have one more question over here.
A comment, reaction.
A comment or reaction, yes. Okay.
I feel this discussion, by the way, I'm Chun Li from the University of Michigan. I think the discussion has been so far sobering, talking about difficulties and just negative and sad. I want to make some positive comments on a great deal of good things that are happening. For example, the 3 principal platforms, gene panels, 500 genes, when you design them well, do recognize 30 to 40 cell types in a tissue section with 150,000 cells. We may well have 100 or 200 functional units, and then you can pseudo-time them, you know, the spatial proximity. So that's really great. But I want to come back to another question. How do you summarize from this local spatial domain character to a sample-wide single statistics that you can compare, say, 10 disease and 10 normal? A lot of these are context-specific. Involves us to invent new metrics to discover, to describe spatial characteristics. I already heard something about neighborhood large or small. For wound healing, we talk about the jaggedness of the edge or some type of spatial entropy of mixing, whether immune cells go mixing or stay together. These different descriptors can be quantified and summarizable either locally or to a sample. So a lot of the, just like in bulk RNA, we talk about differential expression. With single cell, we talk about 2 things. One is the right cell cell number, and another is differential expression in known cell types. With spatial, we talk about whether they're together in the right place, whether these units are misshapen or in kind of right size or not. So I think that's my last comment.
Yeah, maybe that presents a really nice to like segue actually to discussion B as well, because like if I heard you correctly, like what you're saying is like you would like some sort of like method or way to identify metrics that you could actually then relate to the phenotype or an outcome in sort of like with respect to like your samples or whatever data that you're analyzing. Is that a correct interpretation?
Right. We talk about say immune infiltration, or whether the rigidity or extracellular matrix are this or that. We can score them with spatial data, including we don't measure force, but the cells are elongated, wise eccentricity measure, and can talk about magnitude patterns of the cell.
So maybe it's like a question back to that or to the whole room. Like, are there specific properties or features that you think can only be obtained by spatial omics data and not from image-based data? Because I think it's like we should be very careful when we're applying like the spatial omics methods. But they are more expensive, right, and requires more effort. So if you can delineate the same information from an image, maybe it's like more appropriate to use an image. But like maybe you have like some examples, or like it's open for discussion. Like, do you think there are specific inherent properties of these tissues that we're studying that requires actually spatial omics data?
In the example I know best is the tubule structure in the testes where inside the sperm is made. So the geometry of the cylinder, you have genes and cells distributed radially outside in and longitudinally along the tubule. Once you establish the norm in the healthy adult fertile male, then you have then you quantify the variation from the norm. You're in a great place to study what happens with aging, with different types of fertility or subfertility. Likewise, if you go to the ovary, where the geometry is here, the distribution of cells and genes are outside in. So then, once you know the norm, variation from the norm, you can score infertility and premature ovarian aging and so on and so forth. These are not possible with the dissociated cells.
Yeah, just briefly and then I'd like to comment. Just throwing in there, I think like in a lot of ways, that is then almost like a common coordinate framework, I would guess, like you characterize the tubular structure, and then there's like a notion of distortion of that. Yeah, and exactly here, I think it sounds like you have an idea of how to build that framework for a bunch of other program organs. I think it's, or maybe other length scales, it's not always easy yet how to capture them best to then see those perturbations. Yeah.
So this is Ray Chen from Breakout Session. So we work on the retina. So just want to share a little experience on what we learned from the spatial data on the retina. It's not served by single-cell RNA-seq. So the first thing we find is that the single-cell RNA-seq is way activity faster cell types, but there are cell types on the RNA dimension that can separate. We notice cases with, for example, some admin cells. They are at two distinct locations, different layer, and they have different morphology, they have different function. But when you look at the transcriptome, they only differ during movement, so it's not distinguishable at all. But if you have the spatial data overlay on that, actually, and then so that's one example. So there are cells are not defined or easily defined. So the second one is cells seems very similar, like the photoreceptor cells are very similar transcriptome level. When we do the spatial and we did some imputation, we find there's a gradient across the spatial position. And that we don't understand biology significance of that gradient, but we think they are correlated with the cell generation, the function, metabolic rate, things like that nature. So there are things that we cannot discover with DNA. So, but related to my question or comments, one thing we find difficult is to find those unless you have the whole transcriptome, spatial data, right? So we rely on imputation. So we basically combine this RNA-seq with spatial, we impute. But when we try different imputation methods, they give different answer. So we were able to validate maybe say 30, 40%. We pick a handful of genes that we find gradient or something. We're able to validate maybe 30, 40%, we say our scope. But the other 50% is not exactly. So I'm wondering whether there's any we benchmark, or improve the imputation until one day we can need it.
Maybe so. One idea on that could be, for example, that I think right now when you're doing like your panel design, essentially, a lot of the panels are optimized for you being able to distinguish different cell types, but maybe not to actually allow you to impute the gene expression. So maybe you should like change the objective when designing the gene panels, trying to find like genes that like are highly informative of other genes, like high correlations, like you're designing for capturing the full transcriptome rather than just like specific cell types. I think that could be one way to like approach that kind of problem. And you might like learn those genes, like those gene sets from single cell RNA-seq data, or maybe you could even use full transcriptome, spatial transcriptomics data, like this year more slides. It used to see like what genes it tend to co-express, and then you tend to pick like significant genes that allows you to impute more of the gene expression. And the reason for using spatial omics data when actually doing your gene panel design would be to like also capture some of the biases that you have using a spatial method versus a single cell method. That would be one suggestion, I think, but there are probably different ways of doing that as well.
So in terms of the role of spatial, I have a comment that this is based on very practical consideration. So if single cell seq versus spatial, single cell seq, I mean, this is my impression that for clinical use, it will probably never be practical, because part of the reason is very logistic reason, because to make a single cell suspension from clinical setting is not very practical, whereas using a tissue embedded in paraffin is practical. So I think if going forward, once we start thinking about how to apply this human cell atlas, it will necessarily be spatial. So in terms of use cases, I think that's probably one important consideration, just because it's more effective.
I think we had a question in the back, maybe in the corner, but maybe that was answered.
Jasmine, and it goes back to your point about the cell imputation. And I think it requires us in the field, one, to have more data, but we don't know what the ground truth is, right? And just because we have single cell annotation full transcriptome, that doesn't mean what we're capturing in a more reduced level isn't real. And so I think it requires, we don't have metrics yet, but depositing as much data as possible as a community so that we can allow the computational methods to catch up, right? We're where we were 6 years ago in single cell. And I think it's our responsibility not to force the imputation, because we are seeing things, and this goes back to Giovanni's point. The reason we do spatial, in the sense back to his point, to even need spatial, is we are seeing cell neighborhood. Whatever you call a cell neighbor, no one really, like I call it, it's awful. It really is arbitrary, but I think that's really the cytoarchitecture of what the gentleman just mentioned in terms of looking at the tissue, and we are seeing those things. So to reduce that level back to single cell and say we have to find a one-to-one correlation, we get a 30% dropout from the cell types, but what if the cell types are truly being defined by cell neighborhoods, not the cell transferring of the labels? And I think that's something we all have to kind of think about and just keep as much meta and as much not collapsing of the data as possible so that we know what the ground truth will be over time. But I don't know if we know that yet. That's okay. We all just have to accept that that's okay.
We're kind of like in this positive. So to add to that, actually, single cell suffers from the problem of, again, that long dissociations expressed for a really long time. And actually, spatial, like you mentioned, can uncover much more of the biological variability that is in the native state of the tissue. I see that's one of the biggest spatial, just because you're using an FTP block that was kind of taken relatively quickly processed as opposed to dissociate themselves. So they always feel uneasy about whether we're actually capturing this kind of average cell that is boring and all its like unique profiles, and these things have been kind of dampened out through the hours of processing.
Does the isolation of nuclei have a position? Because
you can do that at low. Yeah. So actually, in that sense, I think that the nuclei, they have the problem of kind of like you get like kind of like less biological diversity. Identifying the same cells in nuclei and single cells is possible if you talk about integration issues that as well. Actually, in that sense, like the platforms that use fixed cells for dissociation, like Flex or Parse and other platforms, I think are really exciting. I'm sure someone is working, like, as we speak, kind of like comparing these mediatory genes structured like a function using tissue. But
We have a paper out, so whoever's researching
Okay.
it. So we have a single nuke FFP Flex. Really working through like
Yeah.
fixed cells.
I mean, that's
And we think it's important. It's not
Yeah.
full biology, but it's important.
well, that's the most important thing. I always think that I always like find these gems in PhD works of people that really think that it's not important research things like this. I think it's critical. So yeah, so to your question, these conversation methods are special.
That's awesome. So I think we've kind of like hung out here at Discussion A for quite a while now, and it's super essential that we actually consider like how we're generating our spatial omics data, because it's kind of like what we usually say, we're like shit in, shit out. So like we should be careful with like what we generate, but we also think it'd be super nice to just like move on maybe towards Discussion B, is like focus a bit on the computational methods for the analysis part, is like maybe try to assess where you guys feel like the most need for like new methods or like improvements of existing methods might be. So if anyone has like any comments on that, it would be super interesting to hear, and maybe you could share that with the audience. Yeah.
So in terms of deconvolution methods, I'm just wondering, once digital spatial becomes more widespread, what would be the use case for investing more on developing these methods? I mean, other than using existing data, how would it benefit?
Yeah, that's a super good point. Then like 5 years ago when I submitted my paper on this, they asked the exact same question. So I guess you're really on point there. And I guess it goes back to like using archival data to some extent. Like it seems like people here are presenting like a lot of Visium data. People are investing in generating it. So it's still nice to kind of like have a method to analyze that. But maybe, as we said like earlier on, like this might be one of the topics where we shouldn't invest more and more time and effort, but maybe sort of like try to settle on like what the state of the art methods are, and then try to refocus our efforts on some of the other topics. But I'm definitely not the one who's gonna say this is what we're gonna do or not do. But I think you're raising a really valid point there, especially with like the advancement of new spatial technologies that are single cell resolution. So maybe someone else wants to comment that.
Okay, so say you want to do a genocentric analysis. So, I kind of want to get a sense of people's opinion. Do you think, so we do need convolution to we're guessing the cell type, basically, that's going on. But, say you want to say, you're doing cell-cell interaction, and you say, in this spot, you have gene A, and in the newest spot, you have gene B, and in the third spot, you have gene A. So you get that kind of pattern, something like that. So we don't need deconvolution. Do you think that's like a reasonable thing to do with the kind of slide C sort of data, or you still want to deconvolve it somehow at the cell type level and then do something?
I think it depends on the system. So if you're these like tens of micrometer slot sizes, right? Like there's definitely signals that live on that area, like a bunch of diffuse factor gradients, like neutron gradients. But yeah, then there's these other biologies where like it would definitely like I guess like I think like a lot of immunology would be centered around like actual cells touching, and then you might want this finer resolution, but it's also just a comment actually of comment. Yeah. So, reader.
I just wanted to comment. So I'm from 10x, but I wanted to comment on the single cell versus spatial and kind of how we're thinking about it. And I actually really love other people's thoughts. Like So also the thing when you look at like single cell right now is like the strength. I know people think of it as expensive, and in some ways it is, but it's also actually very efficient compared to spatial methods at sampling many samples, right? Because you can go in and you can take a large collection of samples and run many cells. And I think with some of the innovations that we're trying to drive to, you can like, hundreds of dollars a sample, generate lots of data on lots of cells. And the other advantage for single cell versus the single cell spatial methods is the types of analytes that you can measure, like the breadth of analytes you can still measure, and the multiple modalities, and then also like the depth of information, like to really go super deep on a whole transcriptome. High resolution spatial is still not, at the same level of information on all the cells in the tissue is still not there with spatial. So, but this is something like we debate a lot internally. So I would actually like, I'm really curious how people react to that, and how they, does that resonate with you guys, or do you feel like, no, like when some of these cases you're just gonna stop doing single cell, you're gonna do, high resolution spatial and not need the reference anymore?
I'm a spatial center, and I will still always do single cell, right? So I just think that I realize, and I'm sequencing based in my mind, and I think everybody in this room is. And so I trust the analytics better. I trust the ground truth that an A, a C, a T, and a G has been embedded for us, independent of the molecular biology we're throwing at it. And I think spatial gives us a way to validate, right? So that's clearly the path for me, that we still use kind of discovery, cell type phenotyping. Now's our job as HCA to kind of put that into disease, or allow other biologists to put in disease and put more samples at it. And then use kind of, I think it's unfair to ask spatial technology right now to be like everything all at once, that everywhere all at once, I think is the theme. I think we will be, but I think we know it's tight. We can throw multiome at it. There's lots of different questions that we still have to answer, and then we can validate, I think, based on the site of architecture that we're seeing.
Before next question, can we just take one question online first, maybe, to be a bit inclusive here? And so, Vitaly, I think you're with us, so maybe you want to just like read your comments or say what's sort of like you have in mind.
Yeah, hi. So, I mean, first is that I agree with using single cell and spatial data, using single cell RNA-seq until spatial data that can reliably provide single cell profiles. And this includes both solving segmentation challenges where that's relevant, solving the cell mixtures challenges, which are still present in sequencing-based spatial data because of how it's done. I mean, it's very, I think that we have to do single cell atlasing until that happens. And I just wanted to say on the previous discussion that sometimes in spatial data, you can observe gradients of populations that are missed in single cell data, but I think it's important to distinguish whether they are missing because of biology or because of technical issues. If your single cell atlas doesn't have certain cell populations because of isolation, or because detection of certain cytoplasmic genes didn't work very well in single nucleus data, it doesn't mean that the same heterogeneity is not present in both modalities. And I think it's important to recognize this difference when thinking about analyzing this data, when thinking about developing new computational methods, just that there is, just that with all of these methods, we're essentially trying to profile the same cells. Hopefully this makes sense.
Yeah, thank you so much, Vitaly. I think you're raising a super good point there about like we need to like try to understand what the inherent biases of the technologies are before making like solid conclusions on the biology side. So yeah, that's a really good point to bring up. Did we have some comments there? Yeah.
To follow up on this interplay between single cell and spatial. In our hand, specifically, it's the Murphy-Shake spatial without single cells. So with dissociative cells, after 50,000 cells, we kind of hit a diminished return without the cell types. We start to litigate with other cell to define those difficult cell types well or not, such as progenitor cell, which might be hybrid or doublet. With single cell, when you have 100,000, 200,000 spread in front of you, you have a lot of advantages. One is the cell census is unbiased, super rare and super abundant ones, or in front of you, let's say in here, can't be. And then the suspicious so-called potential doublets, you can tell, and you can pursue whether it's a segmentation problem. Some oddly shaped cells like to be wrongly segmented. Others are genuine, have no doubt that cell exists. You then can study whether its neighborhood influenced it to become a transitional state. And then in a lot of functional units, spatial gradient is a pseudo time so long you have a differentiation wave or some hormonal gradient. So those are true advantages that with associated cells, you just it's mired into guessing, you do pseudo time, the velocity. You still don't know who is going, who is neighbor of whom.
I mean, of course, spatial data informs. I just felt like I need to reply to this. Of course, spatial data informs all of these questions that you just mentioned, which cells are nearby, that actually gradients in transcription may be associated with spatial access, like villi in the gut. But the heterogeneity in transcriptional states should still be present in measurements from both technologies. And if spatial data enables new questions, but the transcriptomes are there in both cases.
Yeah, so I think we had like a bunch of thoughts on the relation of spatial transcriptomics to atlases, either through like how they cover each other's disadvantages, clinical applications, or in general clinical applicability of single cell. Maybe we have like 5 minutes left or so. Any thoughts on discovery of biology beyond single cell biology in the end? So like more like tissue biology concepts, like where do people like see that going? Yeah.
Maybe I can just add one thing to that before we go to the question. And so one thing that is kind of like unique to spatial data is kind of like these cell-cell interactions. So I can imagine how that's like really relevant when you're sort of like trying to use a spatial technique. And so it would be super interesting to also hear it's like what your thoughts are like these cell-cell interactions and what you think sort of like constitutes a good like proxy or metric for cell-cell interactions, because it's like the one thing that's like really hard to get from single cell data. Even though you can do some sort of like inference of the cell-cell interactions, here you can actually observe them to some extent as well. So I think sort of like incorporating that in the discussion would be super nice within these like last 2 minutes, I guess.
This is more of a question, you know, for those who have used both single cell seq and spatial, you know, can they share their experience about single cell meaning, you know, nuclear seq or single cell seq? What is the type of concordance among the data? Is it so when we go forward, you know, most of the human cell atlas probably will be built from single seq data, and when you want to translate it to spatial, do they correspond really well? Do they not? Or, you know, the fixation techniques and all that, will that create a major difference, batch effects? If anyone has that experience.
I can share some thoughts. So we saw today in the morning the Cytex example, right? They have extremely high concordance with associated single nuclear sequencing because it's so similar essentially the way it's processed. I think with panel-based techniques, you have what would be considered very strong batch effects compared to like what you encounter within single-cell RNA sequencing. But often you can recover structures using projection techniques. But if you were to look at individual features, there's pretty strong differences. Yeah. But
so will it be possible to merge these data types?
Yeah. I think yeah. There's a few works looking into this, and then it's all about like up to what level of granularity can you merge? Up to what level of granularity do you believe the merge? But yeah, I think we're going to see this now because the atlas is only now at that level of granularity, and we don't have so much spatial data yet, but yeah.
Follow up on your question on sense interaction very quickly. Like I think that one of the things that we can learn from neuroscience is that actually when you see neuroscience is known like maximum dendrite that are co-located in space, the probability of them forming connections to be around it. There's no inference in that sense. You cannot predict anything from the fact that they're co-located in space. So much. So and I actually get the feeling that also in cell biology that might also hold. So the fact that two cells are proximity together might be more to the fact that one is more abundant than the other in that specific location, because it's probably like a cytokine gradient, but it has nothing to do with the fact that they're directly interacting. And you actually, and I would suspect that you maybe would need to have to image the neurological synapse in this case. And I think what would be kind of really useful for future spatial technologies is to specifically probe and like have these hybrid models, right, for protein and RNA, when you kind of like can you have the sense interaction and you also have the like the immunological synapse staining that you can say that indeed these two cells are interacting with one another. And I think it will really help untangle.
Thank you so much for that comment. I think that will also like act as a really good evaluation data set, right, for any method that we're to develop that only relies on one of them. So I think we're kind of like on time. I'm gonna wrap up the discussion. Thank you to all of you who engaged, and yeah, super happy to continue discussing these things later in the poster session or anything, right? So thank you.